Good afternoon, everybody. Now we are going to start the knowledge sharing session of the National Dengue Control Unit. Today's topic is an update on dengue vaccine. I'm very much thankful to Dr. LakKumar Fernando, consultant pediatrician, District General Hospital, Nikambu, readily agreeing to deliver this lecture on behalf of us. Now I call upon Dr. Himali Herat, consultant community physician of the Dengue Control Unit, to proceed from here onwards.
Thank you, sir. Good afternoon to all of you. Welcome to the knowledge sharing session of the National Dengue Control Unit. For today's session, we are privileged to have Dr. LakKumar Fernando, consultant pediatrician, District General Hospital, Nikambu, as our guest speaker. Let me introduce our speaker, Dr. LakKumar Fernando. Dr. LakKumar Fernando is a senior consultant pediatrician, and currently he is also the clinical head of the Center for Clinical Management of Dengue and Dengue Hemorrhagic Fever, Nikambu, Sri Lanka. The Center for Clinical Management of Dengue and Dengue Hemorrhagic Fever, which is his concept, is the world's first dengue treatment facility that handles both children and adults with dengue fever under one roof, together by a common team of physicians, pediatricians, and other staff. And the center has received international recognition. Dr. LakKumar Fernando is a key contributor to Sri Lanka's national guidelines on dengue in children, adults, and in pregnancy, and a contributor to the latest WHO Zero guideline on dengue. Recently, he was appointed to the WHO Regional Technical Advisory Group for dengue and other arboviral diseases in Southeast Asia region. The Ashok Foundation of the United States awarded him the Senior Ashok Fellowship in 2012, recognizing his change-making role in dengue. And in 2016, the Asia Pacific Pediatric Association, representing over 27 countries, awarded him the Outstanding Asian Pediatrician Award. In 2018, Dr. LakKumar Fernando received the highest category of civil awards of Pakistan, conferred to any Sri Lankan national from the President of Pakistan, in recognition of the service rendered to Pakistan during its major dengue epidemic in 2011, and for the impact created in saving lives from dengue in Pakistan. He has many publications and presentations, both national and international, to his credit. And he is a well-known invited speaker at many international conferences on the disease dengue. Last year, he won the New Investigator Award from the International Society for Infectious Diseases for his research on fluid therapy in dengue hemorrhagic fever, submitted to ICID. Dr. LakKumar Fernando is also the coordinating principal investigator of an ongoing international dengue vaccine trial, and he will be discussing this too at today's lecture. Dr. LakKumar Fernando, sir, over to you.
Thank you. Thank you very much for inviting me to talk today. I'm sorry about the fact that we had to postpone this meeting due to other commitments and another meeting that we had before. I'm of course a clinician, but my main topic that I generally discuss is about the clinical management of dengue, but I also happens to be a principal coordinating investigator of a ongoing dengue vaccine trial in Sri Lanka by the for the Takeda vaccine. Also, I have served the Asia board in another dengue vaccine for a short period. Coming back to the dengue virus, I suppose a lot of people are quite aware about the viral structures these days, having studied the COVID virus. I would just briefly show that the dengue virus has, this is the structure of the dengue virus. It has a structural and a non-structural protein areas. We have the envelope protein, the membrane or the perimembrane, and also you have the capsid, and inside you get the non-structural protein. There are 7 non-structural proteins, which we of course know the NS1, which we sort of use for the diagnosis. And this virus has 2 sort of regions, what we call untranslated regions 5 and 8. And in this viral structure, that the vaccines are developed using various manipulations of the viral structure, so that the most important thing about the dengue virus is, unlike the COVID, that we have 4 viruses that really need to be handled. And if you get immunity, we really need to bring immunity against all 4 serotypes. So there are these 4 serotypes which are sort of have different we have different they are different at amino acid levels 25 to 40%, and also all these 4 viruses can still produce the same disease. The symptom-wise, severity-wise, there is you cannot say one virus type is more severe than the other one. And each infection is known to give lifelong immunity against the same serotype, and some cross protection against all 4 serotypes for a short period. This is also sometimes disputed. There are very occasional cases where you reported somebody developing the reinfection with the same type rarely, but then medicine is like that. And now dengue virus is unique and complex because it is unique and complex. Development of vaccine is also not easy. If you really look at the COVID vaccine development started in a very flash, and it went very fast, and we've got already some successful vaccines. But dengue vaccine development has happened long before, but we still haven't got good success. So the main reason is that we need to protect against all 4 serotypes. So the vaccine has to be tetravalent. And the other important thing is that you need to have protection that will last long. Now, one important observation, and it is not always mentioned in books, and also I think with our experience of dengue, we probably have experienced the same thing. The dengue virus, when you get infected, you can develop infection 4 times during lifetime. But the most important or the most serious infection or the worst infection is when you get it for the second time. But the only thing is you don't know when you get it, whether you get it for the first time or the second time, because your previous infections can be asymptomatic. So first infection is usually mild, and the second is the worst. And an important observation is 3rd and 4th are usually milder than the second. I think as a clinician who is practicing, I also have noticed this. There are people who have got it second time, and then they come when they get it for the 3rd and 4th time, and we have often seen that they are not as severe as when they get it the second time. Then the key feature here is that if you give give a vaccine to an individual, and the vaccine can act as the first infection. And so as a result, for a vaccinated person who hasn't developed a proper clinical infection as such, when he gets infected for the first time, then it can be his second infection. It can act as his second infection. So that there is a theoretical possibility that if you are not protected after your vaccine, you have already got the vaccine, which means you have got the first infection, you're not protected, therefore you got the second time. So the second infection, or you get the first wild infection. The first wild infection itself, which is otherwise going to be mild, is going to be severe because you have had the vaccine before. That's the problem. That is what is sort of known as antibody-dependent immune enhancement. This is that if your body is circulating antibodies, which is either passively transmitted. Sometimes it can happen from a mother to a baby, and to a young infant, where you have got the antibodies, but if they cannot neutralize and completely protect, then those antibodies can facilitate the entry of the antigen into the cells for bigger replication, and then produce bigger viral load and bigger cytokines as a result. This is the example. When you get the first attack, the dengue virus will just get into the antigen presenting cell, and inside the antigen presenting cells, it'll multiply, and it will produce the more virus, but at the same time, it will also produce a degree of, you get all these B cells, T cells that produce your antibodies also. But when you get a second attack, what would happen is that there will be some heterogeneous antibody. All the antibodies which are specific for that will neutralize the antigen or the virus which is the same type. But if the antibodies are not good enough to neutralize them, then what will happen is that they will be binding, and then they'll facilitate entry into the cell, and as a result, that you will be producing increased amount of the virus, as well as all the chemokines, cytokines, as a result of this thing. That is what is known as the cytokine storm. Now, this was postulated that it would happen even in the SARS-CoV-2, but I would think that this is a slide that I took from something last year, but we haven't seen this yet. But whatever it is, we really don't know. It can take time, because depending on how long your effective antibodies can last in your body. Now, vaccinating the population. When you want to vaccinate a population, you need to really understand that we get all kinds of people in the community. There are people who haven't ever been exposed to the dengue virus or the seronegatives. Then you get people with the past infections with just one type of the dengue virus, just first infection only. And there are ones who got dengue infected with dengue more than one time. Of course, many could be, or all could be asymptomatic. And then there are other flaviviruses like Zika, JE, yellow fever, and so on. And there can be people who are infected with those, and who carry antibodies against those also, which may be cross-reactive. So those are the problems about the development of a vaccine against dengue. And still people don't know how the immunoprotection against dengue is set at. There is no sort of, you cannot get a antibody titer and say that this amount of antibodies, if you can measure, that it will protect. There is no such correlation. And then also there is no antibody titer for sort of specific serotype. Then one of the main reasons behind the dengue hemorrhagic fever or dengue shock syndrome, you get a transient period of leaking only in some individuals, is attributed to the severe dengue producing all the cytokines that produces capillary leaking due to the immune enhancement phenomenon. But then there is also T-cell mediated cytokine storm, complement activation, all kinds of things happening. But there is no clear understanding as to a mechanism where we could sort of go to one end and stop that. Then there are instances where people who will get dengue only for the first time can also develop, especially I've seen it in dengue 3, where you will develop dengue hemorrhagic fever, though there is no mechanism to explain immune enhancement where you have been infected before. Then there is also no proper animal model to describe dengue or the study dengue. You need there to use either immunocompetent mouse resistant to clinical effects of dengue virus, or what you call non-human primates, or human volunteers to study the dengue virus and the dengue vaccine. So this is a list of course, I'm sure that with the current experience from the COVID vaccine, that there will be more dengue vaccines to come, more technologies to come. We haven't still, I haven't really come across an mRNA vaccine against dengue as yet, which is seeing any promise. But we can see that these are the dengue vaccines that are currently at the forefront. What we call the only licensed vaccine is the Sanofi Pasteur Denvaxia vaccine, and which is called CYDT-DV, and it's a live attenuated virus, which is the only licensed vaccine. Then we have the Takeda vaccine, where I said I'm the current coordinating principal investigator for Sri Lanka, where we have 4 sites running: Negombo, Ledrige Hospital, Colombo South, and Colombo North hospitals, where we have included participants for this trial. Then we have another vaccine called NIH, or the Bhutantan Vaccine, which is also now entered phase 3. It is going slowly in phase 3. And these are the vaccines. There are institutes, GSK, Walter Reed Institute of, and so on. There are a lot of vaccines are developed actually in America, and there are some subunit vaccines which are now developed by India also. And coming back to the vaccines in the development, I said because there are 4 serotypes, and we need to develop a vaccine that is capable of giving immunity against all 4 serotypes. The biggest challenge is how to combine them. The combination is done with the called the chimeric vaccines. The chimera is a kind of a term that you are hybriding 2 new microorganisms. They are you just putting things of nucleic acid fragments from 2 or more different microorganisms together to produce something that is necessary for replication. So in the Denvaxia and Takeda vaccines, they are sort of what you call molecular chimers. What they have done is they have taken the structural genes of dengue 1, 2, 3, 4 in Denvaxia and put it into the backbone or the non-structural on the non-structural genes of yellow fever virus vaccines. So they have taken the membranous and the envelope, a part of the structural proteins of those 4 viruses into the yellow fever vaccine, where the non-structural genes are there. Then you get the Takeda vaccine. It's the structural genes of then 1, 3, and 4 is taken and put it onto the dengue 2 virus backbone. So the rest is the dengue 2 virus. But from the dengue 2 virus, I'll be describing it more later. The NIH vaccine is slightly different. They have taken the whole virus of virus 1, 2, and 4, and then they have taken chimera of then 4 and 2. They have taken the N4 virus and put the structural genes of the dengue 2 on it. So this is the graphic representation, and this is the virus structure. And you can see that they have taken these 2 membranous and the envelope parts of these are 1, 2, 3, 4, and the yellow fever viruses. On the yellow fever virus, they have put the dengue, the 4 structural genes of the 4 serotypes of the dengue virus, and the TDV vaccine or the Takeda dengue vaccine. What they have done is they have taken the dengue 2 virus remains as it is, but on that, by removing 2 of the structural genes, they have hybrided the structural genes of 1, 3, and 4. In the NIH vaccine, those are the 3 most important vaccines that I want to talk to. They have taken the virus as it is, virus 1, 3, and 4. They have taken the 4, and into the 4, they have put the structural genes of dengue 2. So that's how these 3 vaccines are made. But there are lessons that we need to learn from this vaccine story. Then if you take the Denvaxia, the first dengue virus vaccine, which was initially, as it came in, it was something very promising. It came in, the first license was given in Mexico in 2015. Currently approved in about 20 countries, but not used in 20 countries as such. And Sri Lanka did not give registration for it in 2017 when it was discussed heavily, because that came in as a vaccine to be given for everybody. We had reasons to discuss. If you have during question time, I can answer them. Then the other vaccine I said is the Takeda vaccine, is undergoing phase 3 trials in not in 10 countries. Actually, it started with 10, but we are doing it 8 countries. And the NIH vaccine has started the phase 3 vaccine in Brazil, and that is as Bhutantan Vaccine. This was developed by the American National Institute of Health, and what they have done is they are given free license to anybody who wants to prepare it. As a result, there are various other countries that are interested in, and see, India, 2 companies in India, including Serum Institute, has taken over to develop it further. I'll tell more later. Now, coming back to the experience that we have with the dengue vaccine, and also the experience as well as disappointment, is the Denvaxia. It was developed by Sanofi Pasteur, starting from 2015, live attenuated chimeric vaccine. They studied the vaccine by using the experience that they had from yellow fever vaccine. Yellow fever vaccine was a good vaccine, and probably with the yellow fever vaccine, they thought that they can make a dengue vaccine easily, but it did not work that way. They used the yellow fever 17 backbone. It tested the safety and efficacy of this vaccine in children as young as 2 years to 16 years in 10 dengue endemic countries using 35 children, both Asia and Latin America, at a ratio of 2:1. That is, out of the subjects, 2 were given the vaccine, 1 were given the placebo. That is how even the COVID vaccines are studied. I sort of not generally you give more vaccine to the people, otherwise people will not come for the trial if they don't have a higher chance of getting the vaccine. And a smaller number, 1/4 or 1/3 is given the placebo. And the results that we just saw was a real setback. So this is the vaccine efficacy of the seronegatives in the first publication that was published in 2015 New England Journal, that showed that the risk of how the seronegatives perform, the significance of in the Southeast Asian region, the vaccine efficacy was only 34.9% in the seronegative people. And in America, it was 42%. And in combination, it was only 36.6%. This is the overall efficacy. In the seronegative population, people who have not developed or had dengue before. Of course, they did a mistake. They did not check the sero status of all the subjects also. They did it mainly in a subset before the trial. Our vaccine trial, which involved the Takeda vaccine trial, had a lot of things to learn from that vaccine, from the Dengue vaccine study. But when they did it on the seropositives, they found that the seropositives means by the time they develop the vaccine, the seronegatives are actually people who are receiving the vaccine as their first infection. So when they develop the disease, it is the second infection. So they develop more, their chances of developing more severe disease is high. If you're already seropositive, when you get the vaccine, you already had one natural infection. So the vaccine will act as a second infection. What you get is your third or fourth. As I told you at the beginning, when you get the third or fourth infection, your chances of developing severe disease is less. So as a result, when you measure the vaccine efficacy in the form of severe disease that you found, the seropositives perform very much differently to the seronegatives, where you get 73% efficacy in South Asia and 83% in America, and combined 77%. Now, again, here, what really happened was what is described as antibody-dependent enhancement by my both friend and teacher, Professor Scott Halstead, long years ago, sort of came into real action. Of course, I've discussed with Professor Halstead, who was predicting this very much. He said definitely this is going to happen well before when this trial results are published. And what they found is in the young group, when you're young, your chances of exposure to the virus is very low. As a result, they found that in the 2 to 5 year age group, when they vaccinated as say 1,636 children in the vaccinated group, there were 15 children who had to be hospitalized compared to only 1 child in the placebo group out of 800. And of course, the numbers are 2 in the vaccine group and 1 in the control group. But even if you adjust to it, that you get when you are getting 1,600 out of 15 out of 1,600 are getting hospital admitted despite getting the vaccine. There is only 2 out of maybe the same number will get admitted. Out of 800, only 1 get admitted in the placebo group. So the placebo people who did not get the vaccine was doing better, because when they develop the first illness, they get milder disease. Here, when these people get it for the second time, because the vaccine is their first infection, so they get more severe disease that need in hospital admission. They saw the same thing in the 4 to 5 year age group. The numbers were less. So overall, when 20 were getting admitted in the vaccinated group, there were 2 in the control group. Even if you adjust it to equal the numbers, 4 to 2, 4 to 20 is the number of people who got. So there were people who developed were given the vaccine suffered more after the vaccine. And when they looked at the efficacy against the different serotypes also, they saw that for dengue virus type 2, they used different formulations of the vaccine, and they found that the efficacy was relatively very much better in dengue 4. But when you go by serotype, with dengue 2, their efficacy was very much less. So that was a unfortunate situation where you get a most promising dengue vaccine quickly getting licensed and disappointing. And so though it was first approved in Mexico in 2015, both WHO as well as the manufacturer had to change its position. And I'm sure it is not impossible for even the current COVID vaccines with time to come to have different but currently what we want a vaccine to quickly protect us and protect us for the moment. But as time goes by, all these things can change potentially. So in April 2016, the WHO SAGE group, which is the strategic advisory group of experts, recommended that you have to give 3 doses 6 months apart. And but they said that if your country is not very zero prevalent, if you have a lot of people who are zero negative, then the vaccine can do more harm than good. Therefore, the endemic countries, if zero prevalence is more than 70 only, that in the more than 9 age group, because of the results that I showed, because below 9 years, your chances of exposure was less. As a result, they said it is not recommended to give this vaccine to younger children below 9, and the company itself took that stand. So they said if you are above 9-year population, zero prevalence is more than 70, you can use the vaccine because you will have an overall benefit. But if you are above 9-year or overall prevalence is less than 50, then you should not be using the vaccine. I'll be showing how the decision of Sri Lanka not to use the vaccine, whether it is right or wrong, going by this, because we also discussed it in 2016. In 2017, November, Sanofi had to sort of come out with their data, and they told the world that there is overall vaccine efficacy against symptomatic dengue. I mean, when we are measuring even with COVID, we are not really measuring the transmission rates or the positivity rates. We are mainly looking at the clinical efficacy of reducing severe disease or symptomatic disease. So similarly, when they looked at symptomatic dengue, they found that there is symptomatic dengue was effectively prevented in the baseline seropositives about 9 years, and with a sort of effectiveness of 76% of the people, but it was much lower among the seronegatives. When you separate out the seronegatives, the seronegatives, it is only 38.8%. They said this on late November in 2017, and they said there is an increased risk of hospitalization and severe dengue in the seronegative individuals starting after 30 months after the first dose. Now this trial, that is after 30 months, that it will take time because there will be cross-protecting antibodies that will protect against all 4 serotypes for some time. And by several months, it will completely go away. So starting from about 30 months, they found after the first dose, these are 3 doses given with 6 months apart. If you count from the first dose, that you can see the seronegative people who have received the vaccine having more hospitalizations. So they concluded that in areas of more than 70% dengue prevalence, they recommended the vaccine, and over a 5-year follow-up, they said that every 4 severe cases, I mean, the vaccine is capable of producing severe cases in the seropositives. So it definitely benefits the seropositives. I said that if you have already got one dengue, your vaccine becomes the second. So the third one is less than your first. Third one is less severe than your second one. So what you get is a third infection. So that way, when you give the vaccine to a population that has more than 70% seroprevalence, every 4 severe cases that you prevent, that you will have one severe case for 1,000 vaccines. If you give 1,000 vaccines to people, that 1,000 vaccines will prevent 4 people from developing severe disease, but 1 person who is seronegative will develop severe disease as a result of the vaccine. Then for every 13 hospitalizations that prevented seropositive, that prevented hospitalization in seropositive vaccinees, that if you give a vaccine, you can prevent hospitalization in, when you give 1,000 people, 13 people will not get admitted among the seropositives because of the vaccine, but 1 person who is seronegative, who would otherwise not get hospitalization, will get admitted because he received the vaccine. So this was a tricky situation. Following this announcement by Sanofi, the WHO had a SAGE group had another look at it, and then they said that you need to give only to seropositives. So now the vaccine's label was changed. I think in Sri Lanka, the NMRA Vaccine Advisory Panel, I was also in the Vaccine Advisory Panel. Somebody would have thought whether there was a conflict of interest. No, I was actually saying that if this vaccine was to be given to people who are known to be seropositive, who has a past history of definite dengue, that it is a useful vaccine to prevent more severe disease later. But the company was not willing to do that. And later on, the WHO said that it has to be labeled as a vaccine only for the seropositives, that you need to do a test. Either you have to have a history of dengue NS1 positivity or serological proof of previous infection, or you have to do an indirect ELISA test, which is a complicated, not very cheap test to test whether you have got past infection or not. And if you have a very high transmission rate, you probably can use rapid antibodies to see whether you have got past infection, but only thing is it's a very insensitive test. If screening is not possible, they recommend to give it only if you have a seroprevalence as high as more than 80%. And I don't see many countries that have such high seroprevalence to use this Dengvaxia. So even after a test, one thing you need to remember is, even if you test for there can be an occasional false positive. There can be a test that no test will be 100% specific. So there can be an instance where you will get the vaccine, but your true seronegative might develop, still get the vaccine. And as a result of the vaccine, you will develop severe disease. So you need to warn the patient that subject that the test is not 100% sure, that efficacy is high for seropositive, but not nil for seronegatives. So Dengvaxia of the future, the Dengvaxia got a place in the epidemic setting. If you have an epidemic, if you have a lot of known people who have had in the past, you probably have a place for Dengvaxia. Because one thing you need to remember is only 25% of the patients are symptomatic. 75% of the dengue cases are asymptomatic. Therefore, it is not always possible to ask everybody to take this vaccine. So if you have past history of dengue with serological confirmed or clinical illness with serology, then probably it is useful. Of course, it will protect you for a short period. Therefore, if you are in a non-endemic country that is traveling to an endemic country for a short period, you can probably use it as a travel vaccine. Coming back to the Takeda vaccine, which I'm also involved in as a coordinating principal investigator. This is we get the red is the backbone of the dengue 2 virus. On that you have put the structural proteins of the other 3, And that's how the vaccine is prepared. It was initially developed by Mahidol University of Bangkok, and it was a live attenuated. And the various parts of this dengue 1 and 2 were brought from various places, say dengue 1 and 2 from Thailand, dengue 3 from Philippines, and dengue 4 from Indonesia to make the structure. And this is the plan of the study, that you had part 1, where you have to, I mean, even for the COVID vaccine, that is how it is done, that you take a number of people are given the vaccine, and after that you follow them up for a certain period until a specified number of cases, a calculated number of cases develop the disease, and then see how many have got the placebo, how many have got the vaccine. So we thought that there will be, the protocol was that wait till 120 cases develop the 120 cases develop the disease, and then unblind and see who are the people who get the vaccine, who are the people who got the placebo, or for it to go on for a minimum duration of 12 months post second vaccination. Of course, all this had to be fast-tracked for the COVID vaccine because we needed the vaccine faster, and you get a phase of active surveillance, which is 6 months, and we got modified active surveillance for the next 3 years. This is the plan of the vaccine trial of the Takeda vaccine, which is trialed in Sri Lanka also among one of the 8 countries. We had the part 1 where we go on for 15 months. You get the first one, and another one, second dose is given after 3 months. And then after they recruited 20,100 subjects, and out of which 2,100 were from Sri Lanka. And the vaccine was given at a 2-to-1 ratio. 13,400 people were given the vaccine, and 6,700 people were given the placebo. And then there were efficacy points at 15 months, 21 months, and so on. And then looking at long-term efficacy. And we published the data in the New England Journal of Medicine in 2019, November. And 26 sites in endemic countries, 53% in Latin America, and almost half of in Asia and Latin America. Sri Lanka, 4 sites, as I told, in those 4 places. And the first dose between September 2016 to March 2017. And I said the Sri Lanka's seronegativity, it was 38.5%. So we had actually almost, our seroprevalence is about 62%. So we are not a qualified country to receive the Dengue vaccine according to the second recommendation, if you really look at. So that's what we got the figures as our seronegativity when we started the trial. Comparatively, we are like Thailand and so on, but countries like Philippines, they have had this disease for decades, and as a result, their seronegative population is relatively small. So this is the overall picture: how many were, what is the age group, the sex, which regions, how many in the placebo, how many in the arm, vaccine arm, and the Sri Lankan scenario. I'll tell you a little bit. And by the time that we went to when we found, we went through the big dengue epidemic of 2017, which of course skewed the results of this vaccine trial to some extent. Of course, it showed more efficacy. And he gave it out of the 122 cases that occurred in the 8 countries and so many sites. Out of the 26 sites, only 4 sites for Sri Lanka, but 68 were large number of them were from Negombo. 68 out of 120 cases were from Sri Lanka. And also they happen to be mostly dengue 2. This is the Sri Lankan scenario. We screened 2,721 subjects, but we recruited the 2,100. We failed the ones whom we thought we cannot follow up. And then out of the 2,100, we selected 2,096, except 4 they received the first dose, and 2,077 stayed in the study to receive the second dose. And so these are the age stratifications of the subjects in the Sri Lankan sites, how we recruited subjects. This has gone on for many years now with sort of a very good retention. At the end of part 1, this is the overall efficacy of the vaccine. Out of 20,071 participants, at least who received at least 1 dose of the vaccine, there was 94.8% received both injections. They were according to protocol. The overall vaccine efficacy and safety of the population was 80.9%. And 78 cases per of the 13,000 subjects, the vaccine group, but several more in 199 in the placebo group. If you really, placebo group was half. If you check 13,000 people, so it would have been 78 against 400 cases vaccine versus placebo. So in the protocol analysis, the vaccine efficacy was 80%. And that was, when we looked at the seronegatives, 61 cases in the virologically confirmed dengue in group and 149 cases in the placebo group. So 27% of the per protocol population was seronegative at the baseline. It showed vaccine efficacy of 74%. Unlike the dengue vaccine, there was no major difference between the seropositives and the seronegatives. Then there were 20 cases of virologically confirmed dengue in the vaccine group, whereas 39 cases in the placebo group. It is almost again double, because this 39 should be 80, because the number of placebo was half of the recipients who received the vaccine. So incidence of the serious side effects were similar in the vaccine group and the placebo group. And this is the chart that showed a clear difference between the incidence of dengue, the virologically confirmed dengue. This is virologically confirmed dengue, and this is hospitalization. In hospitalization, it is shown even better that it is there's a huge difference between the people who got hospitalization. So it is not only that it prevented the virologically confirmed disease, but it also prevented severe disease. But when you looked at the serotypes, of course, we had a problem that we showed more efficacy in the dengue too, because Sri Lanka having a major dengue epidemic at that time, and we did not have many cases for dengue 4. So this was what it is. When it comes to the number up, you can just see that dengue 4 cases has been in many countries. The numbers were low. Dengue 1 and dengue 2 were the sort of the main one, but one we were able to detect during this period. Of course, in Philippines, there were more dengue 3 cases, fortunately, so that we could just look at dengue 3 as well. But the overall dengue 4 was less. This was published, that 24-month thing was published later in the Lancet. And this is the same thing. And but as you say, that this showed that we really need to study this more with regard to go on for a longer period to study this more about the efficacy, with the serotypes. So when we look at that from that point of time, that is that there is some evidence that the onset of after the first dose, there was 80% efficacy during a period. And between doses, the results support the potential benefit regardless of the previous dengue exposure. So there was no difference hardly any difference between the seronegatives and seropositives. And for outbreak control, if there is an outbreak, this vaccine can come and help both the seropositives and seronegatives. And also it can be used as a trial vaccine. We looked at, there is a way in which when you're doing a trial, that you look at what you call serious adverse event. Every hospital admission is considered serious side effect. So as a result, the current position is that we are thinking of we decided to give a booster. And actually the booster also has started out of the Sri Lankan scenario. Out of 2,100 who received the vaccine, already 1,429 are eligible for vaccines, and we are already giving the booster vaccines. We have seen 104 dengue cases in those patients subjects who received the vaccine. We don't know how many are exactly placebo and how many are, give you some details of how many are the whole unblinded thing, and I cannot be telling you. And I also don't know, except to an extent that it has been published. So we get subjects admitted in 2016, 17, various number of admissions. 2017, mostly it is dengue or suspected dengue. And when it comes to hospitalizations, most of the hospitalizations were sort of trivial and completely unrelated ones, but we had 104 subjects admitted with dengue, because in Sri Lanka, we admit all the dengue cases, unlike in the other countries, whenever they are, because though it is not part of the protocol, Sri Lanka does NS1 antigen test in almost all patients, definitely the subjects who were as a standard of care in the country. The dengue vaccine trial, my general belief is this can be the best vaccine, but unfortunately still coming out very slowly. You get the dengue virus 1, 4, and 3 taken, and on 4 that you have put structural proteins of 2 form this vaccine. What they did, they used a live attenuated tetravalent dengue vaccine, which is genetically attenuated, and they took dengue 1, 3, and 4 viruses and put it into this. What they did was they examined several monovalent vaccines. They used one monovalent vaccines and used the monovalent vaccines to see how good the vaccine is, each vaccine is with regard to immunogenicity. And then they use more technology where they removed 30 nucleotides from the 3 UTR region and did some other modifications, and then put them together and tried it from different formulas to produce good seroconversion and produce more neutralizing antibodies. In that, they found that when they give it to a population of sort of healthy volunteers, were given the vaccine, and when they were challenged live attenuated sort of strain of dengue 2 and dengue 3, they showed protection. The solid immune response, and which gave boostering effect after 12 months of the initial dose. So that was and also there was good T cell response, and a single they want to bring it as a single dose live attenuated vaccine to give a sort of maybe a booster later, and monospecific dengue 1 to 4 neutralizing antibodies, which are equally good against all 4 types of noticed. So they published their phase 2 data, but they are still to complete and come with the phase 3. And what happened in Brazil was that in Brazil there was a major outbreak of Zika virus, and as a result of the Zika epidemic that they have, their dengue is replaced by Zika, and so they don't have enough cases to sort of complete this trial as fast as they wanted to be. And there is this vaccine is taken by the Serum Institute through a free transfer of technology, but the Indian authorities wanted them to do phase 1, phase 2, phase 3 all, and not only phase 3. So as a result, the Indian trial is also not going to come very fast. So theoretically, this could be the best dengue vaccine to come, because it has 3 NS1 proteins. That's the important thing. You know, the NS1 is because they are taking all 3 viruses. NS1 looks like what is known to produce severe disease, and it has 3 NS1 proteins of 3 dengue serotypes. So as a result, maybe that it is more capable of producing immunity against protection against severe disease. These are the other vaccines which I will not touch, but I have also a thought whether we will ever be able to produce a very good vaccine that will protect against all effects of dengue and be completely dengue free is still not very near. But one other thought is whether we can produce a vaccine using something like a vaccine against NS1, which may be we still not fully confirmed, maybe the culprit that is producing a lot of vascular effects and shock and so on. And it is, NS1 is produced during cellular infections, which is dengue virus. And I mean, it is, instead of remaining cell bound, it circulates in large quantities in the blood during the acute phase. And NS1 concentration in serum is in early acute phase is usually showing some correlation with the severity of the secondary dengue infection. And also NS1 interact with certain receptors in the macrophages, monocytes, endothelial cells, and so on, and detecting that during leaking phase. So if there is a possibility of in vitro NS1 neutralization, or to have something that can act against NS1, maybe we can produce a vaccine which will not completely stop dengue, but it probably whether it can produce severe disease. So questions with regard to the vaccine in the future are, will there ever be sort of long-lasting protection from one vaccine against dengue? We still don't know how fast an effective, long-lasting one, that is whether you could complete a vaccine and stop there, or whether you will have to keep taking boosters. And will it ever be a single dose? Like, it is very unlikely that it will be a single dose. If so, how often do you have to give the boosters? Because we are now got into a booster, knowing that there is some waning that is happening after our vaccine trial is now 5 years old. Then whether you could use booster of one vaccine in combination to a different vaccine, like the way that we mix and match the COVID vaccines. Then what about, as I said, something that can prevent DHF, if not completely dengue? Then another thought is the use of monoclonal antibodies against dengue, which is, it looks good, but it is also a dangerous idea. Because one is that if you have no way of clearing the monoclonal antibodies after neutralizing the virus that you give for, whether those remaining antibodies, I know that against COVID that we are giving monoclonal antibodies, we still don't see the remaining antibodies doing any harm because another infection. But whether dengue can do the same is a big question about using monoclonal antibodies against dengue. Then one of the other important things that we noticed is dengue is severe only in certain parts of the country, around the world, in Africa, and especially in the Black population, ancestry population. There are a lot of dengue, repeated infections, but there is hardly any DHF. What is the central protection it gives? And what are we to learn from COVID vaccines in the development? I wish to thank Professor Chitra, who was my teacher on this, as well as Professor Halstead, who introduced the immune enhancement theory to the world, as well as who has been my friend and teacher. Thank you.
Thank you, sir. We have a few questions posted in the chat box.
Yeah.
One is like, if the vaccine after the first infection is considered as the second infection, does that mean those who receive the vaccine will get severe side effects?
Side effects means it is like this. When you get dengue for the first time, you normally get mild disease. When you get for a second time, it is very well observed, known that you get severe disease. So if you get the vaccine, despite getting the vaccine, if you get a second time, that means despite getting the vaccine, if you get the wild disease, that means the vaccine has not been able to protect you. So therefore you get the disease. The first disease that you get is your second disease. So since all your first infections are milder, second infections are severe, in the people who got the vaccine, their first infections are actually the second infections. Therefore, they are likely to get more severe disease after the vaccine, if the vaccine cannot prevent you from getting the disease. It is not side effects of the vaccine. The side effects is a wrong term. If you get despite getting the vaccine, if you are not protected, and if you still get dengue, then you get severe disease. It is like that. That is, now, if you take a COVID, when you get the COVID vaccine, if you get the COVID, you get milder COVID. But in dengue, you have a theoretical risk for people who have not been exposed before. Their vaccine will act as their first infection. Their natural first infection will act as their second infection. So they will get severe disease. It's not a side effect. You cannot call it a side effect of the vaccine. It is a consequence of the vaccine.
Thank you, sir. So there's a question. What is the reason for the second infection to become severe and the third and fourth being like milder than the second one?
Exactly not known. If you really go in terms of what you call the immune enhancement theory itself, one can even think why not it will become worse and worse, but it has not been the case. It is mainly you can have explanations, but it's been an observation. That is probably you have, when you get exposed to a virus, that you have just one type of what you call monotypic antibodies, which are capable of neutralizing the next one. So there is only one, if that is also not capable of doing it, and it can only bind and facilitate entry, then it produces severe disease. Whereas if you have got it more than once, maybe you have, there is no proper immunological mechanism that can fully explain this, but it has been observed that people who get a second disease, second time, is the one that who get severe disease.
Thank you, sir. There's another one. Are there any trials on anti-NS1 medication to treat severe infection with dengue virus?
No proper anti-NS1 medication as such, to my knowledge. One is this monoclonal antibody. The monoclonal antibodies, even I have been invited to be part of a monoclonal antibody, but I discussed it seriously about whether there is a place and what are the pros and cons, including a process called Tazod also, and we thought it is tricky. There is no NS1, anti-NS1 treatment as such, agent as such.
There's another question. Will it be easy to convince the public with this much of confusion? Vaccine as first infection and the natural infection as the second infection will give the severe disease. They're asking, sir, I think practically how we can roll out.
It is of course, when it comes to rolling out the NIH vaccine that will come later, as well as the Takeda vaccine that I talked about, we have shown clearly that there is no difference between the efficacy of this, or there is no difference between the seropositives and the seronegatives. So therefore, unlike the Dengue or the Sanofi vaccine. So with Sanofi vaccine, yes, it is a problem, and as a result, there has been many problems in the country that used it, especially in the Philippines. And Philippines used this in the national immunization program because they had a large number of dengue. You see that the seronegativity is only 12%, and they get repeated infections, so they used it. But later on, a lot of people thought that those people who developed the vaccine developed more severe disease, and there was a big confusion. The government stopped the vaccination. Not only they stopped the vaccination, they even sued people who took that decision to vaccinate, include it in the national immunization or do mass vaccination. Immune enhancement, we really don't know how, because it happens even in the Dengue vaccine, it happens 30 months after the vaccine. So all the vaccine that we are using against even the current COVID, though it is an unlikely possibility, but none can be ruled out. We'll have to observe this for years and see what happens if there is another introduction of a similar virus, but how the vaccine recipients will behave. But we have to first escape the current ongoing threat.
Thank you, sir. I think with that, we come to the end of the session. And we would like to thank Dr. LakKumar Fernando, consultant pediatrician, for accepting our invitation and being our resource person today. We know, sir, it had been a very long day for you. Thank you very much for your commitment. And we also highly appreciate that you the support that you always render to the National Dengue Control Unit. And our special thanks goes to Dr. Prashila Samaraveera, National Consultant, World Health Organization, and Dr. Indika Vidanage of WHO for scheduling this Zoom webinar. And then we also thank our participants for being with us in this webinar. And we look forward to having more webinars in the future. Thank you all. Have a nice day. Thank you.